Welcome back to my channel Biochem Bioworld. In today's video, I'll be discussing the basics of the Lac Operon, that is lactose operon, and the structure of the Lac Operon, how it's working, a little bit of that as well. Okay, so basically, Lac Operon is responsible for the transport and metabolism of lactose in E. coli and many other enteric bacteria. The principal source for the bacteria is usually the glucose, but when the media in which the E. coli is growing, or the environment in which E. coli is growing, and in case in the lab if you give lactose in the medium instead of glucose, it starts metabolizing lactose for its energy. Okay, lac operin allows for the effective digestion of lactose when glucose is not present through the activity of the enzyme beta-galactosidase synthesized from the lac Z gene. Okay, this will be clear in the next slide, what's written in that statement. And next point, gene regulation of the lac operin was the first genetic regulatory mechanism to be understood. It is an example of prokaryotic gene regulation. Okay, you will get an insight how the genes in the prokaryotes is regulated. Okay, I have made a video of a polycistronic mRNA, that is the the prokaryotes usually synthesize a polycistronic mRNA, that is an mRNA which is responsible for synthesizing more than one protein, more than one functional protein. Okay, so the polycistronic mRNA, and lac operon is also polycistronic mRNA, and this is the structure of the lac operon. Okay, and usually an operon, it is having three things. Okay, it is having constitutes the three things. First is the promoter, the operator, and the structural genes. We can say promoter, the operator, and the structural gene, that is pose. Operon is consist of these three parts. Okay, so there is a promoter, okay, and there is an operator, and there are three genes which are under this promoter. Okay, that is Z, Y, and A. Okay, Z is responsible for synthesizing galactosidase, whereas Y is responsible for synthesizing the permease, and A is synthesizing an enzyme, that is trans-acetylase. Permease, it creates a pore in the membrane. It creates a pore in the membrane to transport the lactose from the media inside to the inside of the cell. Galactosidase is responsible for breaking down of the lactose so as to use it as a source of energy. And the next important part which is regulating this operon is the lac I. Lac I synthesizes a repressor. Okay, it is synthesizing a tetrameric repressor which goes and binds to the operator. Okay, when lactose is not present, okay, lactose is not present, I do not require the metabolizing enzymes of that, so this operon is shut down by the synthesizing of the repressor. Okay, the repressor goes and binds to the operator, and the RNA polymerase does not go beyond the promoter region. Okay, so there is no synthesis of the three genes. Okay, but when the lactose is present in the case of media, the lac operon is operating. Okay, the repressor is cleaved off from this operator side, and as a result of which there is a synthesis of galactosidase, permease, and trans-acetylase. That was all about lac operon. Thank you so much for watching my channel. Please like, subscribe, and share.